interesting stuff, don't you think? It is. She's laughing. Yeah.
Because we're watching Modern Family.
Yeah.
And the young girl that's really smart, like Heather here, she goes, they made her use the encyclopedias to do a report because they were going
No electronics.
No electronics there, yes. And she goes, yeah, like you guys would know the difference between a gamete and a zygote. And I said, I know what they
I know the Twilight series, you know, in the Twilight series.
Yeah.
And in the first one, where they talk about anaphase, they're in the microscope.
Yeah.
And I'm like, I know that.
See? So it's amazing what you have added to your knowledge base. You're learning all this stuff and you don't even realize you're learning it.
What?
She watches Twilight.
I'm sorry, me too.
But it is. It's interesting. It's very interesting. It's you. It's your life. You know, and even if this is the only science class you ever take, it's a different way to look at things. It's interesting information. You might be able to get some questions right on Jeopardy that you couldn't have gotten right before. You know, look at the red class half full. Huh? There you go. All right, so back to the science. So, biology and society. Viruses. Who's the World Health Organization? What do they do? Do you know what they do? No.
Aren't they the ones that figure out what the viruses are?
Yeah, they kind of look over different things in different areas of the world and how they spread across. Where does the flu come from? You know, the flu we talk about every year, the shot that you get every year? Yeah, but what? No. No, it doesn't. How do they decide what goes in the shot? Do you know?
Don't they look at the virus underneath a microscope and
No.
just try and different chemicals on it?
No.
Take all this data,
They take all this data.
all this data compiled of who's got what and where and
Global occurrences of flu. And do you know where your flu originates from? Pigs and birds. And it mutates in people. Now where, not necessarily, no, just it's called a coronavirus, the regular flu that everybody gets every year. Not everybody, because, no. Now most illnesses are caused by viruses. So these guys look at which ones are spreading across the globe and which ones might eventually come here. Because where do they start? Again, nobody knows the answer to that question, do they? No. Asia. Why Asia? Because it says it right there. But people live in closer proximity to pigs and birds. So eventually that virus that might be in pig and bird make their way to humans. Not that they don't have very good hygiene, that they live in closer proximity to, exactly. They're very close in proximity to, yeah, but usually you have a barn where the livestock live. It's not necessarily, well, it's not better hygiene, it's better accommodations. Let's put it that way. So the flu and places like World Health Organization and different flus, H1N1, that was kind of came to the forefront a few years back. What's that? Yeah, well, it's again a bird flu, avian bird flu, and how it spreads across the country or through the planet, actually. Now, certain ones can cause more difficulty for compromised people. That's why the flu shot is a good idea. I never thought it was. I was you. until I took a course called Emerging Infectious Diseases. I never miss my flu shot after that course.
How do you know,
It's a combination.
how do you know what exactly is an influenza?
You can actually look it up. There's documentation, so you can always find out what's in your flu shot. Yep, there's an MSDS. When you went and get your flu shot, yeah, they'll tell you what's in it. And again, based on statistics taken by the World Health Organization, it's a combination of a couple of different strains of flu that they statistically determine could be coming our way. So it's a cocktail of 2 or 3 different viruses. Now, you don't get the flu from the flu shot. But if you understand immunology, you know that when you get the flu shot, what does your body do? Yeah, it works on an immune response. And sometimes, while you're working on the immune response to the vaccine, your guard goes down a little bit with respect to your immune system. And if there's other viruses in the area, they capitalize on that, or may capitalize on that, depending on how efficient your immune system is. So you don't get that flu from the flu virus, but you might get sick from something else. Does that make sense? Okay. Does that make sense? Okay. Yeah, that's okay. I mean, and again, it's usually patients that are in compromised situations, very young patients, elderly, or some kind of illness that they insist. Hospital employees, you're going to be dealing with people that have these flus. You don't want to give them to those patients, so they're going to make you take that flu shot. Yeah.
Liam's pediatrician, because he was premature, still.
Yep.
Even though he's 7, because he was premature, has to have a flu shot.
And again, because if some of these nasty ones come along,
Because he was premature.
I mean, if anybody's ever had the flu, now ramp it up a bit. And that's what we're talking about when we talk about ones like H1N1. So they can cause a lot of problems. So that's good. You have a good immune system. Knock on wood and thank the big guy.
I thank my mom for letting me play in dirt.
To expose you to things. I heard this little ditty on the news the other day. It wasn't the news, it was a radio station. It's going to sound gross, but I'm throwing it out there. People who pick their nose and eat it have more efficient immune systems.
Ew, and I give Liam crap
Just saying.
every time he's doing that.
No, but get exposed to things in your environment. That's why little kids, when they're first exposed to different things in their environment, tend to get sick more while they're establishing their antibody base.
My mom never gave us any.
Yeah, and it has a lot to do, not just,
She made us just live through
yep, best thing to do.
Yeah, my father
No
used to
low grade
talk-
temperature, leave it alone. It's there for a reason. So, each year in the United States, over 20,000 people die
Is that more
from gun infection. No.
I think it is.
Oh, this? Yes. That number?
Why
Yes.
are we outlawing the flu?
So, in 1918, and this was the big kahuna of flus, to 1919, this deadly pandemic killed 40 million people worldwide in just 18 months. And again, that's why I get my flu shot.
Yeah, but wait, the 20,000, is it older people that their body
And it's just because the flu was so different strains. So different strains are going to wreak more havoc. And every once in a while we get kind of like the super flu, a really strong, virulent strain. I hate that word. Yeah, and that's what they were worried about with H1N1. So, when we talk about flu viruses or viruses in general, we're talking about nucleic acids. We're talking about DNA. We talked a little bit about mutation in this class, right? Okay. Viruses, because they replicate so fast and so often, have the ability to mutate at a very fast rate. That's why the flu shot you have to get every year. How come? It changes. The virus changes. It changes enough so your body's no longer able to recognize it, and it can make you sick. So DNA, RNA, nucleic acids in general are extremely important to understand. Understand, A, the way cells work, why cells do what they do. So DNA as a chemical in cells was discovered around 19th century. What's that 19th century? What are the years? In the 1800s. Way back with Mendel and his peas and all of the stuff we talked about when we talked about Mendelian genetics, we understood that whatever these bits of information was, stored genetic information. And then we copy this information as a cell and pass it on to other cells. So we all get that, yes? But in order to study the molecule, we have to understand what it looks like, how it's shaped. And that's what the field of molecular biology looks at. It looks at the molecules that make up cells. So the biological study of molecules. So that could be another profession that you could get into. Anybody like genetics? No. Did you find that stuff we just did kind of interesting?
It was interesting. I don't think it lived
You know. So DNA and RNA, we know these guys already, right? Nucleic acid, what are they made up of? Nucleotides, which consist of what? Yeah, a phosphate, a sugar, and some sort of nitrogenous base. If we talk DNA, what are my nitrogenous bases?
Adenine, guanine, thymine, cytosine.
Correct. If I'm RNA,
Adenine, uracil.
Uracil and stuff.
uracil. Yeah. So the order of these, and I keep referring to them as Legos, because it kind of keeps it all straight in my head. The order of these is information. So a nucleotide polymer is a polynucleotide. Poly means what?
Many.
Many. So when I talk about DNA, I got a whole bunch of these stuck together. Yes? Bless you. Nucleotides are joined together by covalent bonds between the sugars of one nucleotide and the phosphate of another nucleotide. And they form the backbone of the molecule, that sugar-phosphate backbone. Everybody with me?
DNA and RNA are nucleic acids, polymers made of subunits called nucleotides. One difference between DNA and RNA is the type of sugar their nucleotides contain. DNA contains the sugar deoxyribose, while RNA contains the sugar ribose. Ribose has one more oxygen atom than deoxyribose. DNA and RNA are each composed of 4 different nucleotides, which differ in their nitrogenous bases. 3 of the 4 bases are the same in DNA and RNA: adenine, guanine, and cytosine. The 4th base in DNA is thymine. In RNA, it is uracil. The nitrogenous bases guanine and adenine each have 2 linked rings of atoms. They are called purines. Cytosine, thymine, and uracil each have a single ring, and these 3 bases are called pyrimidines. For convenience, the carbon atoms in a nucleotide sugars are numbered, beginning with the carbon atom bonded to the nitrogenous base, moving around the ring, and up to the carbon that is bonded to the phosphate group. The 1' carbon is bonded to the nitrogenous base, the 3' carbon to the next nucleotide, and the 5' carbon to the phosphate group. DNA and RNA are polynucleotides, long chains of nucleotides. Polynucleotides are always assembled in the 5' to 3' direction. A covalent bond forms between the carbon at the 3' position of a nucleotide and the phosphate group at the 5' position of the next nucleotide. RNA usually consists of a single polynucleotide chain. DNA, on the other hand, consists of 2 polynucleotide chains. The two DNA chains, or strands, are oriented in opposite directions and held together by hydrogen bonds between the nitrogenous bases on opposite strands. Because of their sizes, shapes, and arrangement of polar groups, the DNA bases form complementary pairs. Adenine pairs with thymine, and cytosine pairs with guanine. The two polynucleotides in DNA wind around each other to form the familiar double helix. The bases can be in any order, like the letters of the alphabet. However, the base sequence is significant. Sequences of bases called genes encode the instructions for the structure and function of an organism.
Holy crap! I'm not going to make you memorize all that stuff. But if you go on and become a molecular biologist, you'd understand what that guy just said. So the structure of DNA, the order of its bases and pairs, and the shape of it is a science in itself.
In 1953, Alfred Hershey and Martha Chase confirmed DNA's preeminent role in genetics by demonstrating that DNA is the genetic material of a virus called phage T2. The phage, which infects E. coli, consists of a head, sheath, tail, and base plate made of different proteins. DNA is packaged within the head of the virus. When T2 comes in contact with E. coli, the phage attaches to the bacterium by its tail. Next, the phage injects genetic material into the cell. The genetic material directs bacterial enzymes to produce viral offspring. When the life cycle is complete, 100 to 200 progeny phages have been assembled inside each bacterium. The bacterium breaks open, or lyses, and the phages are released. Since T2 consists of only DNA and protein, Hershey and Chase reasoned that the genetic material must be one of the two components, so they designed an experiment to determine which it is. For their experiment, the scientists prepared T2 phages that either had radioactive DNA or radioactive proteins. To make the components radioactive, Hershey and Chase infected E. coli with T2 and grew the bacterial cells in two different culture media, one containing the radioactive isotope phosphorus-32 and the other containing the radioactive isotope sulfur-35. All the phages released from the bacteria grown in the phosphorus-32 medium had radioactively labeled DNA, while all the phages released from the bacteria grown in the sulfur-35 medium had radioactively labeled proteins. In the next phase of their experiment, Hershey and Chase infected E. coli with the two types of labeled T2. They then used a blender and a centrifuge to separate the protein parts of the phage, called the phage ghosts, from the rest of the infected cell. The scientists discovered that the labeled DNA appeared in the host cell, but not the phage ghosts, while the labeled proteins appeared in the phage ghosts, but not the host cell. In addition, the progeny of the T2 with labeled DNA were radioactive, while the progeny of the T2 with labeled proteins were not radioactive. Because genetic material is passed on from parent to offspring, Hershey and Chase concluded that DNA had to be the genetic material of T2. Their results helped convince the scientific community that DNA was the hereditary material.
So Mendel figured this out when? In the 1800s. And who confirmed it? In the 50s, 1952. So over 100 years later.
But Mendel didn't know.
No. He knew there was something there, and he didn't know it was DNA. So this chapter talks about the discovering of DNA as our genetic material. The confirmation that, yes, there was something that parents passed on to their offspring, and that something was contained in DNA. So we have a bunch of nucleotides together. If we talk about DNA, it's a double-sided molecule, a double helix. It's put together based on the chemical composition of those nitrogenous bases. So only certain nitrogenous bases can fit together to form the pattern. Other scientists had to come in and figure that one out. Anybody know who?
Figured out that it was upside down. One side was right side up, the other side was...
That was Watson and Crick's discovery.
They figured that out?
They figured that one out with the help of Rosalind Franklin. She never gets any credit for figuring out DNA. Watson and Crick always get the accolades. But it was something called radiocrystallography, X-rays of the molecule, done by Rosalind Franklin, that got them thinking about how this was put together. They tried it right side up on either side, and it didn't come out right. It was the wrong shape. And that's when they figured out, oh, one's right side up, one's upside down. Okay? So, all of these scientific techniques, amazing work, fun to do, something you might think about. So sugars in DNA are deoxyribose. So deoxyribonucleic acid, DNA. We have the 4 nitrogenous bases, adenine, guanine, thymine, and cytosine. And in RNA, which is a little bit different, what are the 2 differences between DNA and RNA? Well, a couple more than 2 differences. It's single-stranded typically. And what else? U instead of T, uracil instead of thymine, and what else? Different sugar, ribose, instead of deoxyribose. So those are some of the differences. Boy, that would make a good bonus question, wouldn't it? What are some of the differences between DNA and RNA? You might want to write that down. So this is a model of the sugar, phosphate, and nitrogenous base coming together in DNA. So James Watson and Francis Crick determined it was a double helix. But they determined that information based on Rosalind Franklin's work with the X-ray crystallography, the X-ray images she was able to produce, which later on, by the way, was her demise. Because X-rays back then, we understand what we got, but we didn't understand how detrimental they could be to our own cells. So Rosalind's work actually killed her in the long run. But it's her images that got them thinking about, okay, how do these guys go together? And that's when they came up with their double helical model. So again, poor Rosalind risked her life for science, and these 2 guys get all the credit. Exactly. So let's give Rosalind her due diligence. Thank you, Rosalind, for figuring out the composition and makeup of DNA. So
What year was that?
it's in 1950. Is it 53? 53, yeah. So her work was probably earlier than that, probably 51, 52. That's not that long ago. Do we have people alive today that didn't know the structure of DNA? Yeah. It's amazing what we've developed with respect to DNA in that short period of time.
That's all that alien technology.
Alien technology. So, DNA is like a little rope ladder twisted in a spiral. Either side represents a sugar phosphate backbone, and then the rungs of the ladder would be the nitrogenous bases. Again, in certain patterns, in a certain order. The other thing they determined is the twist. Which was the twist that was right? They determined that as well. The whole thing, well, it's showing you twist in mid-motion and then as it goes around. It doesn't twist the other way. No.
So the
They always determine the twist direction as well. Correct. So they complement each other based on their shape. They talked about two terms of, my brain just went dead, purines and pyrimidines. A purine can only attach to a pyrimidine. You can't have two purines or two pyrimidines attaching to each other, because then the ladder would look like this. That wouldn't work, would it? Yeah, no. So they determined that as well. So if we watch this little blast animation, we can see how the molecule is formed. So we have two parallel strands, nucleotides that make up those strands, and then the building blocks of the nucleotides, which are nitrogenous bases, some sort of sugar. And which molecule are we talking about here? That's DNA. So we have what kind of sugar? Deoxyribose sugar and a phosphate group. So certain nucleotides in certain order, again we call them polynucleotides, are going to form our DNA ladder. Again, the chemistry of the order that backbone is made in is based on where a phosphate attaches to a sugar, 3', 5'. So it had to do with the chemistry that was outlined in the beginning. So who holds the two strands together? It is. It's amazing how they came up with these things. Hydrogen bonds. So hydrogen bonds, even though they're weak bonds, are extremely important in holding our DNA molecules together. It's nice that they're weak bonds, because what can I do when it's time for me to make new DNA? Yeah, it's with the help of enzymes, easy to break, unzip, and get the information I need from the DNA molecule. Okay, why is this? Oop. I don't know why it does that. So hydrogen bonds are extremely important in holding the shape of the molecule itself, creating the rungs of those ladders. They help to stabilize the molecule. And this is where we discovered that hydrogen bonds were so important in chemistry, that they were, even though they were weak bonds, helped to stabilize molecules, helped to give them their shape and keep them together. And the reason we can have the rungs of the ladder of the DNA molecule. I know there's a lot of little movies in here, but sometimes when we see these little movies, they make more sense because we see pictures moving. I know. Can you see them at home?
I can. What I have to do is I have to make it a slideshow, and then I can.
Gotcha.
If I do it just like if it's the way it is right now,
It won't play.
I absolutely cannot. But if I make it a slideshow, I can.
Oh, good. So the information is there.
Yes.
That's good to know.
DNA consists of two complementary strands of nucleotides twisted together to form a double helix. In this space-filling model, each atom is represented by a small sphere. The yellow and red phosphate groups make the two sugar phosphate backbones easy to spot. They're on the outside of the double helix. We will highlight the backbones in blue. Now let's look at the crossbars connecting the backbones. Each crossbar is a hydrogen-bonded pair of bases, one from each DNA strand. An adenine is always paired with a thymine and a guanine with a cytosine. The double helix can also be represented by a ball and stick model. For simplicity, a ribbon model is often used, with ribbons for the sugar phosphate backbones.
is what you see in your books. You see these pictures all the time. They're based on chemical organization of molecules. If you take organic chemistry, one of the things you might have to buy in the bookstore is going to be very strange. You know what it is? It's a bag of ball and sticks. And those balls and sticks help us to actually visualize the orientation, the three-dimensional orientation of molecules, and how one little hydrogen bond can make a big difference in the shape of a molecule. And shape is going to determine activity. So this is where Watson and Crick came in, gave us all of this information. I know, blows your mind, doesn't it? So every time a cell replicates itself, it has to make a complete copy of its genetic material. So the next cell, the new cell, can do the same job the original cell did. So Watson and Crick's model for that DNA talked about the order in which we unzip and copy that genetic material. It's very specific. This one doesn't play for some reason. I think it's too big. So you're going to kill me. I have it here though. It's this chapter 10, right? These videos were created by Pearson. And do some of you guys have mastering for your
Every cell in your body is produced by cell division. Before each cell divides, it...
Oh, come on, this did this before. Oops. I prematurely clicked. There we go.
Every cell in your body is produced by cell division. Before each cell divides, it must copy its genetic material in a process called DNA replication. Understanding of DNA replication comes largely from studies of E. coli, bacteria that are found by the billions in your large intestine. Let's take a look at how DNA replication occurs in an E. coli cell. As we zoom in, we see the DNA. At the origin of replication, the two strands of DNA separate, serving as templates for making new strands. The result is a replication bubble. The bubble grows in both directions, forming two replication forks. Let's zoom in on one of them. Many proteins work together at the replication fork. Only some are shown. Here, the DNA is unwound, and DNA polymerases, shown in orange, build new strands of DNA. Original parental DNA strands are shown in dark blue. Newly formed DNA strands are shown in light blue. Because strands in a DNA double helix run in opposite directions, the new strands must be made in different ways. One new strand, the leading strand, is built continuously. The other new strand, the lagging strand, is built in pieces. First, let's focus on the leading strand. DNA polymerase builds a new strand of DNA by adding DNA nucleotides one at a time. Each new nucleotide must pair up with its complementary nucleotide on the parental strand. Adding new nucleotides works the same way on both the leading and lagging strands. Each piece of the lagging strand begins with a short segment of RNA, shown in red. A clamp surrounds the RNA and attaches to DNA polymerase, which builds the rest of the new piece as DNA. When the piece is finished, it is released from DNA polymerase. How are pieces of the lagging strand joined together? A different DNA polymerase removes RNA and replaces it with DNA.
Sometimes we refer to that as editing.
However, it cannot finish connecting the pieces. An enzyme called DNA ligase joins the pieces together. Growth of the leading and lagging strands continues on both sides of the replication bubble until there are two identical DNA molecules. Although bacteria are very different from humans, the process of DNA replication in bacteria is similar to what happens in your own cells.
So why do you think it was easier to study bacteria when we were trying to figure out how DNA replicated? How come? Yeah, life cycle shorter, much smaller amount of genetic material. It's not in the nucleus. All those reasons is why we pick on the bacteria to study DNA replication and protein synthesis. So, where am I? Slideshow.
In principle, copying DNA, a process called DNA replication, is very simple. The two complementary DNA strands separate, and because each nucleotide can only pair with its complement, adenine with thymine and cytosine with guanine, each strand can be used as a template to build a new complementary strand, producing two DNA molecules.
In the cell, DNA replication is a little more complicated, but the principle is the same. For clarity, we have untwisted the double helix. Remember that each DNA strand has a 3' and 5' end, and the strands run in opposite directions. DNA replication begins at specific sites called origins of replication. Proteins attach here and separate the DNA strands, forming replication bubbles which grow in both directions. Enzymes called DNA polymerases move along the template DNA strands and catalyze the elongation of new strands. Because DNA polymerases can only assemble new DNA in the 5' to 3' direction, only half of the new DNA can be synthesized in one continuous piece. The other half is synthesized in short pieces. As the replication bubbles grow, one daughter strand is synthesized continuously, while the other daughter strand is synthesized in pieces. The pieces are joined together by the enzyme DNA ligase. Eventually, all the replication bubbles merge, yielding two identical DNA molecules.
So pretty complex, right? With the help of all these enzymes, polymerase enzymes, we can help to build these new molecules. And the ligase enzymes are going to help stick them all together to create two brand new strands. Are they two brand new strands though? Two brand new. Yeah, they're only half new, right? The other half's from the old stuff. So when we have two copies of genetic material, one of the strands is from the original DNA, and the other strand is the new strand we copy or build to it. Yes?
Here you can see the simultaneous formation of the leading and lagging strands. A DNA polymerase assembles a continuous leading strand while primase, other DNA polymerases, and ligase all work together to make the lagging strand. The enzyme helicase continues to untwist the double helix, exposing more template strand DNA for replication.
So there's a whole bunch of enzymes that work together. We have the polymerase enzymes that are going to help take the nucleotides and stick them together. We have the ligase enzymes that are going to help tack all the little pieces that we form to make one big strand. And then we have an enzyme called helicase. And that's the one that's going to unwind the genetic material for us, untwist it, so we can copy it. Is your mind blown yet? Yep. So the result is two new strands of DNA. The daughter DNA, also a double helix, is half old and half new. Okay? Does that make sense? Okay. So during the process of replication, which we take for granted, there can be mistakes, yes? Mistakes can be made. There's a lot of information. There's a lot of copying going on and tacking and pasting, and I don't know if you noticed the term Okazaki fragments in that little movie that we just watched, but that's what they call the little pieces that are then tacked together. Okay? Again, I'm not going to make you memorize this, so take a deep breath. I just want you to get an appreciation for how complex genetic material is. We can damage it by disrupting the order. Things like X-rays and ultraviolet light can disrupt the order of genetic material, so that when it copies itself, it's copying something that doesn't work quite the same. Enzymes are extremely important. Enzymes are going to help these reactions take place. Basically, what enzymes are going to do is bring things close enough together so they can do what? So they can bond and react with each other to form those strands. So when I'm making new DNA, the name of the enzyme that's going to help me make new DNA is what? DNA polymerase. So when you see that word polymerase, polymers, helping to make larger molecules. So DNA polymerase enzymes help me make new DNA molecules. And they're going to help create those bonds, help bring things close enough together to create the bonds between those nucleotides. They also, believe it or not, can repair damage. Can we repair all the damage we do? No, and sometimes the damage is too much and our DNA can't repair itself. So DNA replication ensures that all of our body cells are going to have the same genetic information. And that genetic information is written in both directions. Again, that was discovered by Mr. Watson and Mr. Crick. Is it going to work? No. Well, it's kind of repeating itself. I'm going to skip these couple because we're kind of repeating ourselves. But see, the origin of replication opens up that replication bubble, and then we go in both directions as we copy. 5' to 3' is a continuous copy. That's, remember when these guys made themselves? The sugars and the phosphate attached at the 5th carbon and the 3rd carbon, right? That's what 5' and 3' meant. And again, because we have to flip one side and the other, we see 5' and 3' ends are different in our strands. So that's basically the chemistry they talked about in the beginning of the chapter. So the copy that is made from the 5' to 3' end is made continuously. And we call that the what? That's the leading strand. The one that we have to kind of make piece by piece by piece, that's going in the other direction. That's called the lagging strand. Why do you think they call it the lagging strand? It's going in the other direction, and it takes longer to do, because what do we have to do? Edit out all those little messenger RNAs that we needed in order to create the little fragments that we then have to remove and stick those little fragments together. So it takes longer, it lags behind, and that's why they call it the lagging strand. So in your textbook, you see this picture on page 177, figure 10.7. It doesn't just happen or start in one place. When I'm copying tons and tons of information, it would be nice if I can start copying it in a whole bunch of different places that eventually lead all together. So we see that many of these little bubbles open up when it's time for me to copy genetic information. And what I end up with from the parental strand is two daughter strands, half of which are the originals, and the other half are the copies of information. And again, it's not going to just start in one place. It's going to start in many places and feed into each other. So we have tons and tons of different enzymes that are working on copying genetic material at any given time. Remember, DNA is the instructions. It tells our cells what to do, and eventually has a role in making us the organism that we are. Is everybody's directions the same? Nope. Much of our information as human beings is similar. We all have eyes. We all have eyes. We all have noses, 2 legs, 2 arms, hair. Well, some of us lose that as we get older. You know where I'm going with that, right? But the different information that we all possess gives us different phenotypic characteristics. Everybody knows what phenotype is, right? What's that?
Physical traits.
Yeah, physical traits. So a genotype is your genetic makeup, and a phenotype is what you look like, right? Different proteins are going to make you look differently. Different eye color, different hair color, different skin color, different proteins. Yes? And remember, those proteins are made based on what? The code in our DNA. So the DNA is going to specify what type of proteins you have. Transcription, translation. We went over this a little bit, and the book kind of beats it to death in this chapter. Yes? Okay. Did everybody understand what we were talking about when we were talking about transcription and translation before? Not everybody's shaking their head. Yes. Well, hopefully by the end of this chapter, you'll understand what transcription and translation is. So the genetic material has to be copied to make new DNA. But the genetic material has to be read to make proteins. We have to read the information the DNA is giving us, transcription. And then we have to translate that information into a what? Into a protein.
Ribosomes do all that, the transcription and translation?
Ribosomes are going to help us with that, yep. Protein synthesis is this one.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases called an anticodon that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
Cool, huh? Yes, sir.
The last animation is Friday. The enzyme was unzipping DNA and creating 2 new DNAs.
Right, because that one was what? This is when I was making new DNA. What's this one?
But it was still using RNA.
Yeah, there's little bits of RNA involved in making new DNA.
And they had to be extricated. But this one here, the DNA is going to zip back up after the RNA is made.
Correct. So, I know. When I'm making new DNA, I'm making new DNA so I can copy the information and make new cells. DNA replication. When I'm undergoing protein synthesis, I'm actually using the DNA. I'm reading the information it contains. I'm not making new DNA. Guess what the enzymes are called that make RNA? They're proteins, but you know what they're called? No, what was it called when I needed enzymes to make new DNA? Polymerase enzymes, right? What are the enzymes called that help me make new RNA? RNA polymerase enzymes. Same thing, except I'm going to have different enzymes making different molecules. Yes? Scary, isn't it? So Watson and Crick, and believe it or not, all of the stuff that you just watched is fairly new to us with respect to science. All of the stuff that you just watched is something that we've researched for many, many years and come up with since 1953. So they didn't know this in 1953. They just figured out what the molecule looked like. And from then, scientists have figured out how it works. Yep. They're pretty close. I mean, there might be some differences. You know, if you look at a biology book or a genetics book 20 years from now, 30 years from now, there is going to be different information, new information, added information. Just like if you looked at a genetics book when I took genetics last year. 1986, I graduated from college, so it's been a few years. Yes, genetics has changed. It's very different. There's more details. There's more that we know about all the different players in creating new genetic material. So the transcription and translation that we talked about earlier involves all of these different enzymes that help us to make copies of material. How they work, a lot of work was done by Beadle and Tatum on mold in the 40s. So they looked at some different molds, again, more simple organisms, studying different strains and how enzymes affected the ability for them to replicate themselves or to make substances that were necessary for them to survive. Showed each mutant was defective in a single gene. So the importance of the genes, the order of the amino acids created by the information that we got from the genes was discovered in the 40s. So one gene, one enzyme hypothesis was discovered. It's been modified since. Function of the gene is to dictate the production of a protein or a polypeptide. Now, a protein consists of a couple of different polypeptides. We talked about protein structure way back when, remember? We said we can have primary, secondary, tertiary, or quaternary proteins. And that's basically what that statement means. Some proteins are much larger than others and have different functions. Like those enzymes that you saw in the little film. Those are very large proteins and have certain functions based on what? Based on their construction, their shape. So when we talk chemistry, shape is important because that's going to determine how we react. So genetic information in DNA is transcribed into RNA, then translated with the help of RNA again into a polypeptide. Polypeptide is another fancy name for what? A protein. And then those proteins, once they're produced, have to be what? We're going to fold them all up. And we saw in the blast animation us producing the proteins, then them entering into the endoplasmic reticulum where they were going to be processed or folded in certain ways so that they can do certain jobs. So, what is the language of a nucleic acid? The sequence of nitrogenous bases, the sequence of nucleotides in a DNA molecule. So any given gene may have thousands of different nucleotides. So when we talk about genetics and alleles and genes, we're talking about thousands and thousands and thousands of combinations of 4 different nitrogenous bases. So DNA is transcribed by producing a RNA molecule. So what kind of RNA molecule? Yeah, messenger RNA. So RNA is then translated with the help of another type of molecule, or the transfer RNA. So you saw in the little blast animation that transfer RNA and messenger RNA fit together very specifically based on what? Shape of what? Or the nucleotide sequence. There's a certain portion of the transfer RNA that can only fit into the sequence on the messenger RNA. We call the sequence of 3 on the messenger RNA a codon. And we call the sequence of 3 that fits into it on the transfer RNA an anticodon. And that's based on the matching up of those nitrogenous bases, A to U in the case of RNA, and G to C. So the result is going to be based on the genetic code, going through a couple of different players, a certain sequence of amino acids that will create a whole bunch of different proteins from the code that's read on the DNA. So that flow of information from gene to protein is based on a triplicate code. And again, lots and lots of research has gone into realizing that it's a triplicate code, not a quadruple or any other larger number. A codon is a triplicate of bases, which codes for one amino acid. And again, the codon is found on what? Messenger. Messenger RNA. So if I pull out a messenger RNA strand, like we saw in the little animation, for every 3 nitrogenous bases, we call it a codon. Now it's going to match up code on the messenger RNA. So our genetic code the messenger RNA. Well, actually it's on the DNA and we copy it on the messenger RNA, right? So set of rules that convert a nucleotide sequence in RNA to amino acid sequence. There's 64 different possibilities with those 4 different nitrogenous bases. 61 of them are going to code for amino acids. 3 of them are what we call stop codons. Wait a minute, we're missing one. There's a go codon too. Now here's the scary part. Look on. No! She's like, oh my God, my brain's gonna explode. On page 10, what page are we on? 180, figure 10.11. Here we see all the different possibilities. So these are a list of what?
Amino acids.
Well, they're a list of nitrogenous base codons. So what are we looking at? The codon what? Yep, messenger RNA. Yes? Its complementary code will hook onto it. So if I look at this codon, CUG, what's its transfer RNA anticodon look like? GAC. You see that? And GAC transfer RNA will be carrying which amino acid? Leucine. It depends. I point this out because some books are written, they give you this chart and they give you the anticodon. This is the codon. So this is what the messenger RNA looks like. The anticodon would be complementary to that. Okay? So these are all the different amino acids. How many different amino acids? No. 20. So if you went and tried to look at all of these, you'd count up 20. There's 20 different amino acids. One called methionine is our stop. Our start. So methionine typically is not found in a protein. It's the amino acid we need to start reading the information. And then we need something to stop, yes? So the stop codon here can be found with a couple of different sequences in the amino acids, in the messenger RNA. So we have a start. We know where to start. Where would I always start reading my genetic information? At AUG, and that's going to be tacked on as a methionine. And where do I stop reading my DNA? It can be 3 different ones. UAA, and usually what we see on the end of a sequence of genetics is a whole bunch of UAA, 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 UAA stops. We know we're done. That's how we know to then dislodge that messenger RNA and send it out for the next step. Okay? So we have a start and we have a stop, and all the information in between in different patterns means something. It's going to eventually equate to some amino acid. What the heck are those? They're little tiny mice babies. Have you ever seen mice babies? No, there actually were a gene for fluorescence was incorporated into their cells. And does anybody know how viruses work? We just saw it, right? They attach themselves to a cell and they do what? They inject their genetic material. And then what's the cell do? It incorporates the genetic material and replicates it. So if I can inject some genetic information into cells and cause those cells to unzip and copy that information, I can inject certain, what's the word I want? Traits, thank you, into cells of different organisms. So they took a fluorescent gene and they incorporated that into the genome, the copy of information in a mouse cell. And look what happened. They have little fluorescing mice. I don't know, I think they last pretty long. Certain incorporations don't work very well. Sometimes the cell won't read them. But this is the basis of genetic engineering. Maybe we can, with the help of certain viruses, inject genetic information into cells that may not be able to make things they should be able to make. Maybe a defective protein or a defective enzyme. If we could inject that material into cells with the help of viruses, we could cause the cells to make that defective protein. See where we're going with this? So transcription. Well, and a possible cure or alleviation for a lot of diseases. Right, so transcription makes RNA from a DNA template, uses a process that resembles the synthesis of DNA strands, similar to what we saw when we made new DNA. What do we have to do in order to read DNA? Yeah, we have to, but we also have to do what? We have to unzip it, don't we? Which we had to do to copy it in the first place. So we see a lot of similarities in protein synthesis and DNA replication. Except with protein synthesis, RNA plays a big role. So uracil plays a big role instead of thymine in making those copies. So RNA polymerase enzymes are the guys that are going to help me make what? RNA. Just like DNA polymerase enzymes were the guys that were going to help me make what? Make DNA. And by making DNA, I mean taking nucleotides and doing what? Putting them in line and sticking them together. So remember, enzymes bring things close enough so they can do what?
Bond.
They can bond, react together. So when I want to make new RNA from RNA nucleotides, where do you get your RNA nucleotides from? Nope. Nope. What'd you have for lunch? Buffalo wings! Margaritas. That sounds good. I love buffalo wings. A good blue cheese, I hope. Oh, good. Buffalo wings are what? They're not buffaloes. It's chicken. What are chickens made of?
Protein.
Not where chickens come from. No, chickens are made of cells just like you, right? Cells that have what? DNA in them, yes? And RNA. What'd you do to your chicken wings? You masticated your chicken wings, exactly. That means chewing in science, by the way. You broke them apart. You mix. Oh, he had margaritas for lunch. That's why he's falling asleep. I'm a little jealous because I could probably use a margarita right now myself. But anyways, so you chewed them all up. You chewed up the cells. You're going to pull them through the digestive system and break them down into their molecular components. Now you see where you got your nucleotides from? Buffalo chicken wings. Yeah. You see? So those nucleotides, when you digest something and break it down, your body is going to break these molecules down into their building blocks. And then they're going to use the building blocks to make the stuff that we need to make, like new messenger RNA or new DNA if I want to make new cells. Interesting, eh? I don't know if this one's going to work. Right, that one didn't work. But transcription takes place in the nucleus during interphase. And that's DNA. And then what are we going to do? What's interphase, by the way? Self-growth. So interphase, most of interphase, the cell's doing what? Not necessarily. Doing what it's supposed to do for a living. So remember the girl eating the donut? What cells are going to do what they do for a living and make that protein insulin for her? The insulin cells. Where are they located?
In the pancreas.
Pancreas. So interphase in my cell life is spent doing what I'm supposed to do for a living. In this case, this particular cell during interphase is making different proteins. That's what it does for a living. You with me? Good. So we're going to make messenger RNA, and then messenger RNA is going to leave the cell. Transcription and translation. Well, you have to go where first? Correct. So rewatch that little blast animation that's on there. That gives us all the parts of transcription and translation. Shows us what's going on with the cell when we make insulin. The first blast animation shows us what's going on when we make new DNA. Yeah, well, cytokinesis is the end of the process. When we're doing what's that other thing? When we're getting ready for what? Mitosis. That's also in interphase though too, right? Remember, that's in the S phase of interphase. So near the end of I do what I do for a living, when I finally decide, okay, I'm going to make another one, during the S phase of interphase is when I'm going to replicate my genetic material. And then what's going to happen? But what's that? What are the steps?
G1, S, G2.
Yeah, but what's after that? mitosis. Yeah, metaphase, prophase, metaphase, anaphase, telophase.
There's five though. There's prophase, and then there's pre-metaphase, and then
Yeah. No, it's pre-prophase. Yeah, we don't, yeah. Just remember prophase, metaphase, anaphase, telophase. And don't forget, we have to replicate the organelles too. So what else is happening in there? Near the end of anaphase and during telophase? Cytokinesis. So, ingest that. That was a lot to ingest. Yes, it was a giant mouthful. Go back and look at it again. Okay? And